biology. I wanted to briefly note that the same thing that we have just done now within SimBiology can be done with another piece of software called COPASI. So the COPASI simulation environment is a piece of software meant to import SBML files and meant to do simulation very much in the sense that we did them with MATLAB SimBiology. So what we do, I go through this quickly just as a demo, not assuming that you're doing it with me. If you just follow what I do here, the file has an import functionality where you can get the model. So you import it from the model database. And if I just use another model to have some different output, if you use that SimBiology model number 42, the Nielsen model, you will get the exact same graph that we found just a few minutes ago with SimBiology. If I include another model, the model Kuma 2000, if you put in Kuma 2000 into the biomodels search window, you will get it and can download it directly from there. Also a model of an oscillatory process, in this case of calcium signaling. Clicking open will, with some warning here, import the model just as previously in MATLAB and will give you some information about it. So I don't explain all the details, but I can show you that unfolding the navigation bar on the left-hand side. You can look at the reactions that are implemented there, and you can see a couple of reactions, in this case related to G protein, membrane-bound G protein function. That's a G protein coupled to a receptor that downstream translates into calcium signaling. And there's a couple of parameters which we see in the parameter overview. We can have all the numbers similarly to what we did previously with our parameter for the flow and the reaction parameters in the glycolytic model. And you have, just like there, the initial conditions of all the variables. And in this case, it turns out there's only 3 variables. So it's a very simple model. Okay, so it's very similar. You have the information that you need. Under tasks, you can specify, and what we did previously was a time course. So if you wanted to run a time course, and I just take a little bit more time, say take 100, in this case seconds, for simulation, you might want to run this right away. One thing in COPASI is have to define that you want to see a plot, because you might also do numerical analysis only. But if you define that you want to have a plot and you click on run, immediately you get the output as a graph. And I enlarge this in order for us to be able to see what the outcome is. I can take away 2 of the graphs to just look at, say, the G alpha protein concentration. So that would be simulation of the membrane-bound G alpha component that is free. So that is sort of representing the activity of the receptor. This receptor is assumed to have been activated. And it shows an oscillatory activity which has been observed experimentally in hepatocytes under ATP stimulation. And the reason I've chosen to show this model, if you just try to remember what this graph looks like, this is a simulation of the reactions as they would go on if it were a purely deterministic world, as if there was no noise at all and no impurities. COPASI, and to some extent also SimBiology, gives you the additional option of changing to a stochastic simulation. It means of changing a situation where not everything fulfills the assumption of this perfect deterministic limit. And we're going to see later in the course what that means in mathematical terms. But to say we want to have something that is a so-called stochastic simulation. So that's a different kind of models. For those interested in it, SysMIC Module 2 does a proper introduction into stochastic simulations as well. It's a bit of an advanced topic, but it's important to see that here you can just by clicking that you want to have a stochastic simulation method, you can pick to have this specified as an option of the simulation environment to provide you with information about what the predictions would look in the case where you simulate a not so perfect world. And I think I can just press run and it will produce an output. If I make the window a little bit bigger and again restrict our graph to the receptor active G alpha unit or the dissociated G alpha membrane bound unit, what you can see is that the graph has certain features similar to what we had before, but in other senses deviates quite strongly, in particular deviates that now we're no longer able to see this very clear periodic repetition. So this is very important because, of course, in an in vivo environment, this more stochastically modulated output is much more what we would expect to see when we measure concentrations. And it's indeed, there's also a lot of research going into this, it's indeed much more like what we see when we do measurements. And of course, the idea of doing research into this is we want to find out how is that, if that's a signaling pathway, how is this noisy behavior decoded further downstream. If signaling means you are going to switch off, say, gene expression regulatory proteins, or you're going to somehow affect protein levels in general, then we need to understand how such a noisy signal, which is the realistic signal, how is this being understood by downstream molecules and interpreted such that the information conveyed from the first messenger, the hormone in the blood, via the membrane-bound receptor and its G protein, how is that understood in terms of gene expression when such a temporal profile is being transmitted. So you can see just by this very simple implementation, importing a model that you can download from a database, you have a very convenient way of starting to think about such processes, namely of thinking about temporal fluctuations not as a nuisance, but as something that occurs naturally and that most likely has some information content in it, namely this temporal structure is being interpreted in the cell. So this brings us just concluding that when there is possibilities of having temporal structure, then there is a new means of looking at the emergent phenomenon. This is an example of such an emergent phenomenon as something that is important for the understanding of the functionality, namely in this case of signaling pathway, in the previous way of the regulation control of a metabolic pathway, and there's of course many other associated questions related to it. Finally, a word of caution. A word of caution is in place because what I haven't really mentioned explicitly is that while these simulations provide some evidence of what's going on, you can see we always show one graph. And if you ask where in the cell is this supposed to happen, from the model point of view, the answer would be in the cytosol. But of course, there is no spatial information incorporated here. We're assuming that this represents overall cytosolic changes. But if you're now interested in several compartments, so how does this translate to activity, say, in the nucleus or within the mitochondria, this is not done in the model as it's set up now. You would have to add further equations to the model, so it means you would have to work on it and expand it if you wanted to have spatial information in addition. So that was just introducing the COPASI environment. It's free to use. You can download it. It works on any platform. It's a cross-platform software, and it's being widely used by people who are not mathematicians and who are not experts to set up their own simulation software competently. So it's an easy start and an easy go, and it has a lot of functionality already built in with it, such that if you want to do something like look at sensitivity analysis, here's where you might do your first simulations. And that allows me to switch off my window. And we've seen that the emergent phenomena, in this case the transition from a non-oscillatory to an oscillatory situation, is of big importance if we assume that some information is contained in the temporal fluctuations. And we have therefore studied a first example of emergent phenomena on a network structure which is given to us by biological pathway databases. Final word. That refers now to one of the PDFs that you can download from the materials webpage. Of course, if you have a model that simulates your output properly, that may not be the end of the story. How do you go on forward to decide whether the model is a good one? Well, you need to do some testing. So what the paper says, that you need to find out what is the actual cause of the oscillatory activity, and this is something that you cannot derive from first principles, but that you need to study case in case. And to cut the story short, in the present model, the Nielsen model, it's stemming from the inclusion, if you take all the complicated things away, of a feature which is called autocatalysis. There is, by combining a certain number of reactions, there's a feature that says if nature works according to this model, then the synthesis of ATP in the glycolytic pathway is actually autocatalytic. You take one and you produce two. So you have two, you can produce 4 and 8 and so on. So that leads to an explosion, to an exponential growth in ATP concentration in the first place. This is one of the possible mechanisms, and it could be an explanation according to this model. But the paper that I have uploaded by Madsen et al. 2005, and if you want, you can download it from the materials, tells you that actually there might be other mechanisms in place that could also explain oscillatory behavior, and that you would actually need to work out which of the two is the correct one. Now, don't go into the details, but if you read that paper, it tells you that one way of addressing this problem of having different models that explain the same phenomenon is to do precisely what we saw in COPASI, namely do sensitivity analysis, checking how a perturbation, so how a manipulation of the model, say changing the expression levels of one of the enzymes in the glycolytic pathway, how that would lead to changes of the regulation and of the oscillatory waveforms in the glycolytic flux. I wanted to mention this because it shows how simulations can be useful to translate back and to ask biological questions by means of that model. And I think that's it for today. There haven't been any questions. One question is, what is the reference to from first principles? So from first principles, I mean, can you, setting up the network structure, logically deduce what is going to happen if you're given that structure? And the answer is no. And the reason why it is no is the answer is no, because apart from having the algorithm of how it works, you have the constants that you need to choose how quick the reactions are, and depending on the choice of constants, or what you are going to do is you're setting certain values and produce different types of oscillatory behaviors. The emergent properties can change, and while you can analyze at what point that might happen, what actually will happen, there's no way of predicting at the current stage of knowledge from the mathematical point of view. So only simulations are the way to predict what you're going to expect to see in an experiment. This concludes today's session. This was the use of two software databases, of two software environments to use models that come from a database. And in the sessions that will follow after the Christmas break in the SysMIC course, we are going to look into the components, into the mathematical and computational components, to actually set up a model ourselves and to understand how we can do the simulations, even programming all the necessary steps, which is actually not very difficult.